this class we are going to talk about cell breakage, issues involved in cell breakages, various techniques that are practiced for breaking cells, and a couple of problems as well. So, why do you need to do the cell breakage? If the metabolites or product of our interest is inside the cells, unlike when the product comes out into the medium, then it is called intracellular material. Okay. Sometimes the material could be not exactly inside, but it could be between the two layers of the cell, or that is called periplasmic. So, intracellular material products are things like recombinant DNA products. The periplasmic could be recombinant DNA again. Extracellular could be like alcohols, acids, antibiotics, enzymes. They all come out into the extracellular medium. So, there is no problem if it is an extracellular product. All you have to do is, after the fermentation, remove the cells by centrifugation or filtration, and then start processing the liquid. Whereas if it is an intracellular or a periplasmic material, then what we have to do is we have to harvest the cells. That means we need to collect the cells, not the liquid medium. We collect the cells and then we have to break the cells to recover our product. So, we have to be very careful, especially when we are breaking the cells. You may be generating heat, you may be generating some unwanted metabolites, or you may be affecting the pH. So, the product, if it is a biomolecule like an enzyme or a protein, it should not get denatured. So, there are many techniques available for breaking cells: mechanical technique, thermal technique, enzymatic technique, and I will show you some of those techniques, and they are very important nowadays, because many products may be inside the cells rather than being outside in the extracellular medium actually. So, what do we have to do? After the fermentation, we have to harvest the cells, that is generally done through centrifugation or a filtration. Throw the liquid out, collect the cells, break the cells or disrupt the cells or homogenize the cells. Then you again use a filter, remove the cell debris, dead biomass and so on, and then get your metabolites in the liquid in the downstream, and then you take it down, do whatever you want to do. You may use a membrane process, or you may use a liquid extraction for concentrating, and then you may go into chromatography for purifying. So, these are the extra steps that are followed if the product is an intracellular material, okay, unlike an extracellular material. So, how do you select the technique? There are, like I said, it could be a mechanical, it could be a chemical, it could be a thermal, it could be enzymatic. How do you decide? So, does the bacteria have cell wall? What are the components present in the cell walls? How strong is the cell? For example, plant cells may be very fragile, whereas certain yeast cells could be very strong, the cell walls. Okay. How much heat is liberated? Because if it is a thermally labile, then when you generate heat because of the disruption process, the product could get denatured. Do we need any solvent or any other chemicals? Because there are chemical methods by which you can break the cells. What is the cost involved? Capital cost, operating cost. That means how much chemicals or consumables or heat or cooling I need to have, that is the operating cost. How efficient is the process? How easy is the process? How much waste I generate? Because I will be generating a lot of cell debris, right, and then unwanted intracellular material, they are all waste. How I will recover the product after removing the product from inside the cells? How am I going to recover? Once I have done a cell disruption, because you are liberating a lot of intracellular material, the entire broth could be non-Newtonian. That means it could attain certain viscosity, which may be difficult to process. So, I need to understand that. Am I releasing toxins when I am doing the cell disruption? I need to understand that. If I am releasing toxins, then the intracellular product may get denatured because of the toxin. So, you decide based on all these various criteria. Okay. Now, if you take bacteria, all you know, there are two types, right? One is the gram positive, other one is the gram negative. Okay. They are separated based on the gram staining which is used. So, gram positive bacteria remain stained by crystal violet after washing, whereas gram negative do not remain stained after washing. So, obviously, they have two different types of cell walls. Okay. So, all the bacteria have cell membrane where the oxidative phosphorylation occurs because they do not have a mitochondria. Now surrounding the cell membrane is the cell wall, which is very rigid and it protects the cell from osmotic lysis and other physical processes actually. Okay. So they could be made up of polysaccharides, glucons, mannans, chitins and so on. So there is a cell membrane and then after that you have the cell wall. So the difference between the gram positive and the gram negative is the presence or absence of the cell wall. Okay. So in gram positive, the cell wall is made up of a peptidoglycan layer. Now this layer is very thick, whereas in gram negative bacteria they have something on top of that. The layer may be very thin, but they have something on top of that, that is called the outer membrane. Generally it is made up of lipopolysaccharides, and this is a major barrier which prevents drugs or other toxins entering the gram negative. That is why gram negative bacteria are much more resistant to antibiotics because of the presence of this outer membrane. Okay. Whereas gram positive bacteria have the peptidoglycan layer and they do not have this lipopolysaccharide outer membrane. So the region between the inner and the outer membrane, this is called the periplasmic space actually. So in gram negative bacteria they store a lot of degradative enzymes in the periplasmic space. Gram positive bacteria lack a periplasmic space because they do not have this LPS layer outside, but they secrete lot of enzymes and they carry out the extracellular digestion actually. Okay, so that is the difference you need to understand. So gram positive bacteria are generally 0.5 to 2 micron in size and they have cell walls of quite small, 0.0 to 0.04 micron thick, and the cell wall is made up of peptidoglycan, which is polysaccharide. If you take gram negative, they are much smaller, 0.5 to 1 micron in size, and the peptidoglycan layer is very thin. It has got a periplasmic space, and on top of that there is a LPS layer also, membrane also. They are mechanically less robust, but they are chemically more resistant than the other bacteria. So if I use mechanical techniques to break the cell wall, gram negative will break faster because the peptidoglycan layer is thinner, whereas chemically they are much stronger than this. So mechanically gram positive is much stronger, chemically gram negative is much stronger. So depending upon the type of bacteria which you try to process, you can go into either a mechanical method or a chemical method. Now yeast, if you take, they are much larger in size. They are about 2 to 20 micron in size and they are very spherical or ellipsoidal, unlike the other one bacteria, and molds are very big, okay, and they are sometimes filamentous also, and they have a thick cell wall. So filamentous, they will create a lot of problem, especially in homogenizers or French press, where you have a very small nozzle, because these filamentous organisms may go and block those nozzles. If you take plasma membranes, they are mainly made up of phospholipids. Okay. Now plant cells, if you take, they are very big. They have thick and strong cell walls, mainly composed of cellulose. They are difficult to disrupt. Whereas cultured plant cells are less robust than real plant cells. If you take animal and plant cells, the size varies between 100 to 1 micron. So you see they are very big. Animals have no cell walls, animal cells have no cell walls, so they can be ruptured very easily in one pass itself actually at about 2000 psi. Whereas insect blood cells will require 15,000 psi, yeast cells will require 20,000 psi, and plant cells will require 40,000 psi. You see, depending upon the thickness or the toughness of the cell wall, you will require different types of pressures to rupture or break the cell walls. So animal cells will require lowest, then comes yeast cells, then comes finally the plant cells actually. So animal cells, they do not have cell walls, they are very fragile and they are quite big, they are micron in size. So growing animal cells in mechanical agitated vessel is difficult. That is why generally animal cells are grown in non-agitated vessel. They are very spherical or ellipsoid in shape. They are eukaryotic cells actually. They are enclosed by a plasma membrane and they contain membrane bound nucleus and organelles. So if I am going to recover products and if they are membrane bound, then I need to collect the membrane and then I need to recover the product from the membrane to which it is bound to actually. Now, breaking of cells, like I said, there are different techniques available. You have mechanical technique, chemical technique, thermal technique, biochemical techniques. Okay. So, you can use any one of these techniques depending upon your requirement and type of cells you are trying to bake, whether it is plant or animal or bacteria and so on. So, there are techniques like high pressure homogenization, bead mill, osmotic shock, thermal method, enzymatic method, chemical method, like you may use chemical detergents, you may use solvents like toluene, urea, antibiotics, latex, or you can also use mechanical methods like ultrasonication, freeze thaw cycling. So, all these are large number of methods available, and some of these methods are for laboratory use and some of the methods are for large scale plant use. So, we will briefly touch about each one of the method. Okay. Now, lot of nucleic acids may be liberated when you break the cells. So, obviously they will increase the viscosity of the broth, and then your broth becomes a non-Newtonian liquid. Okay. If you take heat shock type of method, where you are heating it and cooling it and heating it and cooling it, thereby the cell walls are damaged and broken. It may denature the DNA and RNA present inside, and that will also increase the viscosity. If you use high temperature or if the temperature rise happens because of the breaking of cells, then obviously it is going to degrade the protein or the enzyme present inside the cells. So, all these are issues which one need to think about. So, what is a bead mill? Bead mill is a long tube, hollow tube, where there are a lot of beads present inside. Okay. These beads are made up of ceramic or metal. So, they rotate slowly around 1500 to 2250 rpm. So, the slurry comes inside. So, when it rotates, the beads, the metallic or the ceramic beads hit each other, they also hit the cells, thereby disrupting the cells actually. Okay. So, this is quite a good method for industrial application. We can have cells of about 30 to almost 60% by weight wet solids percentage. So, the release of the intracellular material is considered to be a first order process. Okay. Generally we take it as a first order process. C is equal to C naught multiplied by 1 minus e power minus kT. That is how it is assumed actually. So, the concentration will slowly increase exponentially and reach the maximum value. K is nothing but a constant which determines the first order rate actually. K is sometimes defined as 1 by tau, where tau is the characteristic time. Okay. So, this is a first order process, and generally this type of equation is able to describe the release of the intracellular material. Okay. If you take cell disruptor, what happens is there is a chamber, high pressure chamber, there is a low pressure chamber. So, the cells are compressed to very high pressure and they are introduced through this nozzle and released on this side. Okay. When they are released on this side, they come out from high pressure to low pressure. There is a seat also present, valve seat it is called. The cells hit that valve seat. So many things happen, the cells hit the valve seat, cells move from a very high pressure to low pressure. There is a lot of turbulence and shear produced because of the small nozzle. So, there is a compression that is taking place, so in the small nozzle. So, because of all these reasons, the cells rupture, and so the intracellular material is liberated actually. This is a very good method when compared to the bead mill. So, you are not really destroying too much of intracellular material, unlike the bead mill method actually. The heat produced is much slightly less than the bead mill, but still heat is produced quite a lot during this operation. So, enzymes and proteins are released at various rates depending upon where they are located. So, if the proteins are located in the periplasmic, they are released first. If the proteins are unbound, they are released in one pass. If the proteins are bound to the cell wall, you need many passes through the same cell disruptor to release the protein. So, at various time periods, the products keep coming out actually. Okay. So, again we consider this the cell disruption process as a function of pressure. That means amount of pressure we put in, or the delta P, that is the pressure difference at the high pressure vis-a-vis the low pressure region. Okay. and the concentration of the product released is again considered as a proportional to velocity raised to the power 1 by 3, and the velocity or the throughput is proportional to the pressure actually. And again the concentration release is assumed to be a first order process. C is equal to C naught 1 e power minus k n, n is the number of passes. Okay. So, in the previous bead mill we had e power minus kt, here we are having e power minus kn. So, again it is considered as a first order process, and the k depends upon the type of organism, whether you are using a bacteria or whether you are using a yeast, the k values differ, and the k value will depend upon the pressure. For example, if you look here, we see that k is proportional to the pressure raised to the power 2.9 for the yeast like Saccharomyces, and whereas it is raised to the power p raised to the power 2.2 if it is a E. coli bacteria, right. So, the k depends upon the type of organism which is being broken, and n is the number of passes, and p is your pressure. Okay, so, the release of the protein in the extracellular region we always consider as a first order process like this. So, C is equal to C naught 1 minus E t by tau. Tau is called the time constant of cell release, okay, or e power minus kt, again k is inverse of tau. C naught is the maximum concentration or the amount of active protein present inside the cells, and C is the amount of active protein released in the extracellular region. Okay, let us look at a small problem. So, what I am saying is we are homogenizing a cell suspension to release enzymes. 50% of the total present is released in 16 minutes. Now, if I want to release 90%, how much time it will take? Okay, 50% of it is released in 16 minutes. For 90%, how much time is released? So, the intracellular release follows a first order model. So, we all know what is a first order model. C is equal to C naught 1 minus E t by tau. C naught is your total concentration that is possible, t is the time, and tau we call it as the time constant or characteristic time, and so this is how a first order model looks like. Now, the problem says 50%, so C by C naught is 0.5, and the problem says in 16 minutes 50% is released, so t is 16. So, what do? We substitute into this equation and we estimate tau, that is the time constant. So, very simple. So, we substitute all these and we end up with the time constant of 23.08 minutes. Now, the second part of the problem is how long it will take to release 90% of the total. That means C by C naught will be 0.9, and now we know tau, which is 23.08. So, we substitute that into this equation and we are supposed to estimate this time t. So, when we do that, we get an answer of 53.1 minute. So, for 50% release it takes 16 minutes, for 90% release it takes 53.1 minutes. So, you can see from this problem that as we want more and more percentage release, the time will be longer and longer and longer, because any first order process, as you know, it will go up exponentially and it will slowly keep on increasing. So, if we want to have much higher percentage release, time required will keep going up and up and up and so on actually. Now, there is another cell disrupting technique that is called homogenizer. So, what it does is it sucks in a cell suspension, okay, through this piston, and then it compresses your cell suspension. So, the cells have to move out through these small nozzles. So, during that process the cells get ruptured, okay. So, it moves up and down, this piston moves up and down, the cells are drawn in and then cells are compressed and sent through these nozzles, and during that process cells get disrupted. So, this process is much better than bead mill like process where it is very violent breaking of cells. Here it is very systematic breaking of cells. So, the increase in viscosity is not much, but still heat is liberated. So, what they do is after one pass the contents are cooled and again it is sent back for second pass, then again cooled and so on actually. That is how you handle this type of equipments. After each pass it is cooled. So, again it is a first order process. So, but then if the heat is liberated and it is getting deactivated, then the real curve will look like this because of the deactivation, whereas if there is no deactivation it will look like this. So, the deactivation is generally assumed considering Arrhenius type of behavior, you all know how the Arrhenius equation looks like, right. This is a typical Arrhenius equation, K F is equal to K into e power minus Ed by RT. Ed is the activation energy for deactivation, that is deactivation energy. So, if the temperature is high, then T will come there, your rate constant will go down. So, obviously the cell release will be much lower than what is expected because of the decrease in K, okay, because of the heat, that temperature rise and Ed. So, generally they say about 1000 psi can give you a temperature rise of the order of 1.5, and especially for enzymes and proteins, a temperature rise is leading to deactivation of the product, okay. So, that needs to be considered. French press, this is again a technique which is used for disintegrating chloroplast, blood cells, unicellular organism, okay. So, it disrupts the cellular walls, but it keeps the cell nucleus intact. It produces high pressure and rapid decompression. So, very high pressure and quickly decompress it. By doing that, it is not really damaging the whole cell actually, okay. So, the cell suspension is bled through a needle valve to achieve the decompression. So, the viscosity does not change too much, unlike the mechanical methods. So, this is a good method to resort to. Now, another problem we would like to see, imagine that I have a homogenizer, okay, and it is operated at 200 bar. Now, I connect another homogenizer, another homogenizer of same capacity in parallel. So, what happens is the pressure comes down to 150. So, when it does that, I am asking what is the increase in capacity after adding another. So, is it going to be 2 times? It will not be 2 times because your pressure has come down, and we all know that the capacity is proportional to pressure, right. So, the pressure has come down from 200 to 150, correct. So, that means how much? 25% less, but you have added 2 homogenizer. So, it will be 2 times for 50%. So, 2 into 150 by 200, that is 1.5. Why is 150? Because that is the new pressure, 200 is the old pressure, 2 comes because we have put 2 homogenizer. So, the capacity has gone up to 1.5. So, it has gone up from 1 to 1.5, that means there is an increase in capacity of 50%. So, it will not be double the capacity increase, but it will be only 50% increase because the pressure has come down from 200 to 150 bar, understood. You have colloid mills, that is another method where you have this type of rotating cylinders and there is a small gap. So, the cells are rubbed in that small gap and the cells get broken down. This is very good for tissue based material, you can have single pass or multi pass. It is happening because of the high shear and turbulence that is produced inside between the rotor and the stator here, that is called the colloid mill. You can have osmotic shock. So, what you do in osmotic shock is you move the cells from certain osmotic pressure to another osmotic pressure. So, during that process water may enter into it and it may break the cells. So, osmotic pressure is based on the Van 't Hoff's relation. If you remember, pi equal to RTC, T is the temperature, R is the gas constant, C is the concentration of the salt in molar. So, we can calculate the osmotic pressure for any given concentration of the salt actually. So, what happens here? You have a salt solution, okay, and you have cells inside, water here, okay. Now, the salt solution is in this side. So, obviously, there will be osmosis happening here, and so water will enter the cells, and cells get water enters the cells in large amounts, swells it and allows it to break. And this is a very good method because it does not involve heat liberation or it does not involve uncontrolled breakage actually. So, we can calculate the osmotic pressure using the Van 't Hoff's relation RTC. So, if 0.2 molar salt means I can put C here in 0.2, okay. R is 82, temperature is 27, that is 300 K. So, for a 0.2 molar salt solution I will get an osmotic pressure of 5 atmosphere. So, if you have many salts, we keep calculating osmotic pressure for each salt and we need to add up. So, you can see quite a high pressure that is generated with salt, and this method makes use of the concept of osmotic pressure. Ultrasound, this is generally used in laboratory scale you have an ultrasound generator. So, when it produces ultrasound, cavitations are produced, shock waves are produced, so cells get disrupted. So, heat transfer is a problem, that means heat liberation is very high, sometimes protein may get denatured. It also produces radicals, because ultrasound can produce radicals. So, these radicals could be harmful for the cells. So, generally this is done in batch, and generally this is done in small scale or laboratory scale. It is not done in industrial scale application. Okay. So, that is a big problem here, but it is done regularly in the lab scale. So, you need to use radical scavengers, because it produces radicals which may be harmful for certain enzymes and proteins. Then thermal method, where you are heating, that is called the heat shock method. You heat and cool and heat and cool. So, during that process cell walls break, but the problem is proteins and enzyme can get denatured. Another is lyophilization. You cool it and then you thaw it, cool it and thaw it, and the ice crystals that are formed can break your cells. Okay. This is easier method, because this is a better method, because the protein is not undergoing a thermal degradation, but this is more expensive than the heat shock method actually. Permeabilization with chemicals, you can use chemicals, solvents like ether, toluene, phenyl, ethyl alcohol, DMSO, or chemicals like thionins, antibiotics, catatropic agents, chelating agents, surfactants like triton. So, all these can be used to sort of dissolve the cell wall or completely make the cell wall porous. This is a very good method, because it is very method which does not produce heat, does not produce any harmful products, but then the chemical should not harm the protein or enzymes which you are interested in, which is present inside. So, otherwise chemical methods are quite a useful method, because they are very simple method to use, and they are very mild methods to use. Okay. EDTA is a chelating agent. What it does is it permeabilizes gram negative bacteria. It binds to the cations like calcium, magnesium, which are present in the membrane, thereby it is disrupting the membrane stability. That way it is releasing the internal contents actually. Okay. So, it removes these cations, lipopolysaccharides are disrupted. So, that is a very good EDTA. You also use catatropic agents like urea, guanidine. They bring hydrophobic compounds into aqueous solution. Okay. So, they disrupt the hydrophilic environment of water, they weaken the hydrophobic interaction between the solute and molecules. Okay. So, like detergent, Triton X, you can use catatropic agents like guanidine hydrochloride and so on actually. But then the chemicals could be costly, and then later on you may have to remove the chemicals from your product. So, that may require certain intelligent thinking actually. You can use enzymes as well to permeabilize cells. Enzymes of course can be expensive, so you have to be careful to use cheap enzymes. For example, we can use enzymes like beta 1,6, beta 1,3 glycanase, you can use protease, mannase to destabilize the cell wall or make the cell wall of bacteria porous, so that the internal material are released actually. But then the problem is you need to later on remove that enzyme, that could be another issue we need to consider, because if your intracellular material is an enzyme and you are using an enzyme method to break the cell wall, then how do you remove these two enzymes? So, you need to keep that point in mind actually. So, proteins such as protamine or cationic polysaccharide chitosans can permeabilize these cells. So, mammalian cells can be permeabilized by natural substances like viruses or streptolysin. Lysozyme, this is an industrial process. Lysozyme from hen egg white is a lytic enzyme, and it is used to lyse gram positive bacteria. It is used in commercial industrial scale actually. Okay. So, lysozyme is quite cheap, and it can nicely make the cell wall of gram positive bacteria porous, and thereby allow the release of the intracellular material. Okay. So, main drawback, cost could be a main drawback of this, and then removing these enzymes from your final product could be a drawback. Of course, there are other exotic methods like using electrical discharge method to permeabilize cells, but then they cannot be used in large scale. Maybe in very small scale, theoretical scale, one can think about using those methods actually. Okay. So, with that we conclude the cell breakage. It is a very important downstream technique, especially if your product is inside the cells. So, you collect your cells, that means harvest, then you use one of these methods, either mechanical, chemical, thermal, or high pressure homogenizer type of method, and then you release the intracellular material so that it comes into the extracellular matrix, and then you follow the usual downstream purification and recovery and so on actually. Thank you very much.